Yeah. So something that I mentioned in the presentations is that I started with the differences, or the last week we discussed about QTL analysis and association mapping analysis, or GWAS. And they are important, but the approach of them, it's the same, that is identify regions in the genome that have association with the phenotype. And basically there are two kinds of traits. So there are one kind of trait that it's basically control for major QTLs, and one typical example of that in common bean or intrabean is the resistance to anthracnose that is conferred just for one major QTL. So as in this figure, you have just two groups, one group that is resistant and one group that is susceptible, just because there is one gene, one allele that controls that resistant. Okay, we have a question here. Paul asks, what is the difference between the training and breeding population with relation to the target breeding locations? Okay, Paul, it's because if I understand your question, training and breeding population. The training population is the population where you have the phenotype and the genotype. Okay, so you need to have both kind of data, the phenotype and the genotype, to estimate the breeding values of those lines. And basically you want to estimate the breeding values because what you want, actually what you want to do is estimate the allele, the effect of every single allele, and when you sum or you add all the effects of the alleles, that is the breeding value. And you want to do that because with that information you can do predictions. The breeding population could be bigger, so you can select just one percentage of your breeding population, that it's because I don't know, you just have money to get the genotypic information for 300 lines, but actually your breeding population is 1,000. So you just have to select one portion or one part of that breeding population to get the genotype and the phenotype. Another reason why the training and the breeding population could be different is because you want to test or you want to create models for different environments. So that have relation with the Ronald question. So again, you have your breeding population is 1,000. You have 1,000 individuals in your population. But you just can't afford to plant in different environments 300 or 200 lines. So you planted that training population in different environments. And with that, you can predict the little effects of those environments. And with that information, you can predict the rest or the other lines that you didn't include in the training population, but are part of your breeding population. Okay. It's clear, Paul?
Yes.
Okay, great. Yeah, basically the training population is just one part. And there are different methodologies to try to use or to optimize the selection of the training population. One common strategy is use the pedigree information, because remember the relation between the training population and the testing population, that is, the testing is where you are going to do your predictions. So the general relationship, as closer is that general relation, better are going to be your predictions. So one common strategy to select the training population, it's try to select lines in the training that have relation with a pedigree relation with the testing population. And we can do the same with molecular markers. So with molecular markers, we can estimate the kinship, that is the additive relationship between the lines. And we can also do selections with the kinship. Okay. And yeah, we basically somebody wrote that sounds like ML, splitting data sets, and yeah, it's true. Actually, when we're testing the genomic selection models, what I am doing right now in my research is that I have around 400 lines in my that is my population, where I have the phenotype and the genotype. And to test what is the accuracy of the models, basically what I do, it's split that population 70% as training population and the other 30% as testing population. So basically, I run the models to estimate the allelic effects in the 70% of the population, and I predict the phenotype of the other 30% of the population. And I select that 70% randomly from the population, so I repeat the same process a lot of times. So right now I'm using 100 times, just because the estimation, the allelic effect, again, depend of the population where you're doing the estimation. So that is why it is important to repeat the process, and that just give you an idea about how good is your model. And that is why I repeat the process. Like right now that I am the first like a step of genomic selection, that is assess how good is the model. So I repeat the process a lot of time. In the real world, we are just gonna use all our training population to predict like the new lines. But now that I am just testing how good is the models, I split the population where I have both data. Emmanuel asked that it is good. I'm happy that you have a lot of questions. That there is not a standard rule on the size of the training population and the number of markers to use. How best can one decide the right size and number? And that it's a question that everybody has, Emmanuel. And you're right, it's not a rule, but something that is true. It's as bigger is your training population, better are gonna be your predictions. And that makes sense because as much information you are gonna have to estimate the real effect of the alleles, better are gonna be your models. And also, I think that another reason for that is as bigger is your training population, like the genetic relation, or more lines between the training and the testing population are going to have some degree of genetic relation. There is not a standard number for that. In industry, they use dozens of plants in the training population. In the public sector, as universities and a lot of research centers, we just cannot afford that. And that is why there are a lot of softwares that they are trying to use the pedigree information or the kinship, that is again the additive relation, to try to reduce that number of the training population. Because if the training population is small, that is good, because potentially we can plan that training population in different environments, and we can predict the other breeding lines that we have in the breeding program. But there is not a standard number for that. I have read paper that use 100 lines, and I think that it's a really small number for a training population. But yeah, that depends basically in the resources that you have and the genetic relation that have the training and the testing. And about the markers, that it's another question that depend about, again, what kind of population you have and what is the LD in your crop or in the organism that you are working. Remember, when we talk about meiosis, there are not a lot of recombinations per chromosome. So if you are using a biparental population to do your predictions, that I didn't mention that, but the best population to do, or not the best population, but in biparental populations are gonna, genomic selection has the best predictions. And why? Because the genetic relation between the lines, it's really close. But as breeders, we want to use genomic selection in a more diverse panel. So again, but talking about the biparental population, there are not a lot of recombinations, and you are not going to need a lot of markers, because the idea is that you need one marker in LD with each QTR that controls that phenotype. As more diverse, you are going to need more markers. But in maize, for example, they have found a really good prediction with just 400 markers across the genome, because the LD seems that is high in that crop. In common bean, the LD it's not that high, but I will say that with a couple of dozen of markers in dry bean, it's more than enough. But again, that depends of the LD of your crop. As higher is the LD, less markers you are gonna need. And LD is the linkage disequilibrium. And as more diverse it's your population, more historical recombinations are you going to have in that population. So in that scenario also, more markers are you going to need. But it's something that in a lot of paper, it's a common topic to discuss here, because also you don't want you want to reduce the number of markers as well, to try to reduce, if you can create a chip where you can send your samples to get the molecular marker, so that is going to be more affordable. So yeah, there are a lot of crops that use just chip with 6,000, 10,000 markers. And there are no other programs that use GPS to get more than 50,000 markers. That is my case, for example, in my research. But as we're just trying to test the performance of genomic selection, so it's something that is still we are working on, which strategy works best. But yeah, like the theoretical question is that you just need one marker in LD with each QTR that control that trait. So basically that depends about that. As more quantitative, or as more regions in the genome control the phenotype, more molecular markers are you gonna need, and as a higher is the LD, less molecular markers are you gonna need in that population. And Paul asked about what generation will be ideal to plan your training population. And here, yeah, I have a lot of questions about genomic selection, about the implementation of genomic selection as well. And I think that it just depends on the training of the traits that you are interested on. For example, here in Michigan, what we're doing is that in F6 is the first year where we take the phenotypic data in the field. So in the F6, we take the yield, grain yield, we take canning quality and nutritional value. So we take several traits as a concentration of iron and zinc, because we cannot afford to take that kind of data in early generations, and because in early generations, maybe we are not gonna have enough seeds to take that data. So that is the training population that we are gonna use, the F6. And we're gonna use that information to predict other F6 lines that we are not gonna, where we are not gonna check the same phenotypic data, but also we are gonna use that training population to predict the performance in early generations. For example, we can use the allelic effects that we can predict to increase the canning quality, for example, in F6, and we can predict the same the performance for the same trait in early generations as F3s, because in genomic selection we have to wait until the F6 that here in Michigan we have to wait 3 years to take the first data, the phenotypic data in the target traits. But if we can do predictions in early generations, that could improve the genetic gain of convene, because at the end we are going to remove the lines that seems that they are not good enough for the target trait that we're interested on, and we can use better the resources of the breeding program. Really good questions. And now that yeah, you're really interested in genomic selection, something that you have to think, it's you cannot use the same. I think that I have a slide here that I can use. Okay. So I think hopefully, yeah, everybody, it's so this part is clear for everybody, but I'm just gonna repeat that just to be sure. In the training population, you need the phenotype and the genotype. So you have to have that information to predict, basically to do the linear regression, or if you are using another method to estimate the allele substitution effect, but you need to estimate the allele substitution effect in every single marker. And then with the allele substitution effect of every single marker, you just have to add that effect in all the markers of that in sample that you have, and you are going to have the breeding value. And with that breeding value, that is the genetic genomic value of that lines. But as you estimate the allele substitution effect of every single allele, so you can do the predictions of the genomic value in lines where you only have the genotype. Yeah, so here is that basically we have in this formula, we have the allele substitution effects that are the alpha, and the Z here, it's just the incidence matrix that connect the phenotype and the genotype, and alpha, sorry. So basically with that a formula, we can predict the genomic value of the lines where we only have the genotype, and do the predictions on those lines. Emmanuel asked about genomic selection focuses on predicting additive effects. Yes, most of the most common approach of genomic selection, it's just predict the additive effect because it's the easiest effect or genetic effect to predict. And basically it's a linear regression between again here the amount of alleles that we have in the genotype, for example, here the A1. So each time that we increase in the genotype 1A1, what happened with the phenotype? So that is a linear regression between the genotype and the phenotype. Ana Manuel asks, how can you accurately predict for the not additive epistatic effect? There are some models or approaches, Emmanuel, that try to take into account for non-additive effects. One of them, for example, is machine learning. So that is not a linear model, and that kind of models try to capture like that kind of variation that is not additive. But as I mentioned in the video, they look promising. So they look that makes sense that the models try to take into account different genetic effects, but in the real world, or what has been reported is that those models are not don't have a better performance than the linear or the additive effects so far. So maybe that is going to change soon, but so far the models that are more complex and try to take into account not additive effects and epistatic effects are not outperforms the most common approach, that is the models that just try to capture the additive effect. And again, as I mentioned, although it's true that the additive effects don't capture all the dominance deviation, they can capture, like in this graphic, part of that dominant deviation. And so they are good to capture that dominant deviation as well. So they are not perfect, but they work pretty well. And again, there are several papers that mention that the most important genetic effect in the phenotypes is the additive effect, then the dominant effect, and at the end the epistatic interaction. So as it seems that the additive effect is the most important in the phenotype, so that is why these kind of approaches work pretty well to predict the performance to predict the genomic value. But you are right, they are not perfect, and other models as machine learning could be useful, but still the performance it's lower or basically the same that the traditional additive effect models. Cyprian asks about what is the difference between genomic selection and marker-assisted selection. In marker-assisted selection, basically you use just here. I have the graphic. So this graphic is perfect to explain that. In traditional marker-assisted selection, you need the same information at the beginning. So you need the genotype and you need the phenotype. And with that information, what you are going to do using a biparental population or using a diverse panel, you are interested to find the molecular markers that have association with the QTLs. So when you want to identify the physical region in the genome that have association with the phenotype. That those regions are the QTLs regions. And then with that information, you are going to design some primers that you can use in your lab to amplify those molecular markers. And basically what you want at the end is that, for example, here, you can identify 3 QTLs, so that phenotype is controlled for 3 QTLs, or at least you could identify 3 QTLs in that population. And what you want is run the primers to basically amplify the molecular markers that are in LD with those QTLs, that are associated with those QTLs. And at the end, you are going to run those molecular markers, and basically what you are going to try is to stack or to identify genotypes that have most of the good QTLs that you could identify. For example, here the plant B have 3 QTLs, and that means that that plant is going to have the highest performance in the trait that you are interested on. And then you have other 2 plants. The plant C has a performance in the middle, or the average performance, because have 2 QTLs. And then you have another plant that has the lowest performance because just have 1 QTL. So if this is a breeding program and you are just going to select the best line, you are going to select the plant B because have the 3 QTLs. So basically you just need 3 molecular markers and three primers to amplify those three QTLs or molecular markers. And you can select just with that information. In genomic selection is different because, and traditional marker-assisted selection works pretty well if you just have one major QTL, or as in this example, you just have 3 regions in the genome that control the phenotype. So it's pretty easy design 3 primers to amplify 3 regions in the genome and do the selections. That is easy, and we can do that without any problem in the lab. The problem is that traits as yield, for example, seems that are controlled for hundreds of regions in the genome. So if that is true, if we have hundreds of regions that control one phenotype, that means that in every single region in the genome of those hundreds, it's going to have a really small effect in the phenotype. So if the effect is really small, first, it is really difficult that we can identify those regions. So yeah, it's going to be challenging. And then as the effect of every single marker, it's really small, basically we just cannot use that information in the breeding program, because just suppose that yield is controlled for 100 regions in the genome, and every single region only have an effect of 1% in the phenotype. So if we try to do selection for one molecular marker that have 1%, we have 100 regions, so you only have 1% or control 1% of the variation in the phenotype. So it's gonna be difficult to identify that, because we have variation in the 95%, in the 99 remaining percent of the QTLs that explain that phenotype. So that is why it's difficult to use marker-assisted selection for that kind of traits. But with genomic selection, again, we start with the same kind of information, with the genotype and with the phenotypes. And what we do is run the genomic selection models to estimate the breeding value. So in this case, I am not interested to identify the genomic regions in the genome that have association. What I am interested here is to take into account the information of all the genome and the molecular effect of every single molecular marker that we have in the population. And with that, we can estimate the breeding value. And then we're gonna do selection with the breeding values. So in the theory, if we have just the same example with the 3 QTLs, so the outcome is gonna be the same. So we are gonna select the plan B, because the plan B is the plan that have more QTLs that are a good effect in the phenotype that we are interested on. But again, we are not interested to identify the genomic regions. And the reason, again, it's because in quantitative traits, there are just a lot of regions that have association with the phenotype, and it's not possible to identify all those regions. And that doesn't mean that Dr. Rex Bernardo has a paper about that. That doesn't mean that quantitative traits don't have a major QTLs or QTL that have a big effect in the phenotype. For example, in common being, we have a one in the chromosome 1. Yeah, one QTL in the chromosome 1 that control the architecture of the plant. So that it's important for the yield. But that kind of traits are fixed in the population, because as they have an important or a big effect in the phenotype, the breeder have select that kind of traits or that kind of QTLs, and now those major QTLs for quantitative traits are fixed in the population. That means that we don't have segregation of those traits, and yeah. So as that trait is fixed, the segregation of the other QTLs that have lower effects are the ones that we can predict with the models. Yeah. So I was clear, Cyprian.
Hello.
Hi.
I thank you for the explanation. I have had the difference.
Okay, and I was clear now.
Yeah, you were clear.
Okay, great, great, thank you.
Thank you too.
Um, yeah, and somebody asked about like the differences and between some models, and I have this slide, and again there are different models. Machine learning, now that we have like a lot of information, machine learning is like a common approach right now. It's still there. I think that the room to improve the models or the methodologies. And again, we're gonna use in the hands-on section RRBLAB that are basically linear regressions between the phenotype and the genotype with a correction that basically try to take into account the heritability, because if we don't take that shrinkage, basically the effect of the alleles close to zero. And the reason for that is because if we do just a multiple linear regression, the model is going to explain more variance of the phenotype. So it's going to be overestimate the variance that is explained with the model. So that is why we have to do some kind of correction, and RLBLAB take into account the additive variance or the heritability to correct for that. But here you can see that we have different models in this paper, and all of them are pretty similar. So sometimes that depend of the population, that depend of the genetic architecture of the trait, but most of the models looks, the outcome of most of the models looks pretty similar. Bayesian models, I think that they are interesting because they have a prior, you can set a prior distribution of the effect of the markers, and that makes a lot of sense because there are some traits, as for example quantitative traits in general, as yield, for example, that it's controlled for a lot of regions in the genome. So in that kind of scenarios, we can think that there are a lot of a lot of regions in the genome that have effect. Some of them are going to be have a bigger effect than others, but like the distribution of the effects are going to be kind of normal. So we're going to have a lot of alleles that have effect in the genotype. But there are other traits that are controlled for a lower number of loci across the genome. And in those scenarios, by Bayesian models can take care of that. So for example, if we use Bayes C, you can see here that most of the molecular markers have an effect of 0, and some of the, and just like, yeah, few of the molecular markers have a strong effect in the phenotype. And that works for again, for traits that are controlled for just a couple of regions in the genome. So there are like that, it's an important difference between those models, like the prior assumption of the distribution of the marker effects. But still with that, my suggestion with that is that if you are working in genomic selection, you should try different models, and you have to test if some model works better than another one. If you use different Bayesian models, assumptions, the prior assumptions about the distribution of the effect of the markers, it's different or not in the outcome of the predictions that you are interested on. There is not a clear answer for that. All the papers try to test different models. Yeah, but there is not like a standard model, or actually, I think that the most common model is RR-BLAP. But yeah, I just cannot say that one model is better than the another one. I just can say that they look similar in most of the population and most of the traits. But yeah, that depends on your population and the architecture of your trait of interest. Okay. Any another question? So, and here is the like, I think that this helps to understand, to separate the difference between the marker assisted selection and genomic selection. In marker assisted selection, you are interested to basically identify the QTLs. And with the QTLs, you design the primers to amplify the molecular marker that it's NLD with those QTLs or in those QTLs. In genomic selection, as this figure show you, we are interested to find the effect of every single marker. So here we have 10,000 markers, and we are going to use the information of the 10,000 markers in the model to predict the performance of the lines. So there are some regions that have a huge effect in the phenotype, and they could be QTLs. But again, we are not interested to identify the genomic regions. We are just gonna add the effect of every single molecular marker, and that is gonna be the genomic value of the samples that we are interested on. And something that is really important that you take into account or you think is that this is a really good example of genomic selection. So I think that is kind of a beautiful example. Not all the predictions are, they are like worse predictions. So this is, I will say, a good prediction between the phenotype and the genomic value. But it is really difficult that you have a really narrow correlation between phenotype and genomic value, like this black line that I did here, because that means that the accuracy of the model it's pretty close to 1, and that is difficult to happen because there are a lot of noise that could be into your model. So first we have the environmental effects, and that environmental effect in quantitative traits, it's important. For example, in the heritability for a yield, it's around between 20-something% to 40-something%. So that means that most of the variation is because the environment. And that environment variation, it's because the environment, of course, but also because there are some problems with the phenotype. So maybe the machine that you use to take the phenotype was not that good or was not that reliable because several people taking the phenotypes, and the phenotype was kind of subjective. So there are a lot of variation there, and that it's like in the environmental noise, for example. So the models, it's really difficult to get a accuracy or a correlation between phenotype and genomic values about 1. But what I want to show you is that the advantage of genomic, the advantage of genomic selection, or how you can use genomic selection, is for remove or select the top and the bottom of your breeding program. So if your model is good enough, sometimes the correlation between the phenotype and the genomic value is going to be just 0.5 or 0.4. But if the model is good enough to select the top of the lines, so that is something that you can use in your breeding programs. So that it's important that you take into account this. So genomic selection is really difficult, that it's going to give you a correlation between the phenotype and the genomic value close to 1. I haven't seen nothing like that. But if the model is good enough to use that information to select the germplasm or to keep germplasm in your program, that it's good enough. So that is something that you can use in your breeding program. So if that, if the genomic selection can reduce the money that you need in your program, if can speed up the breeding process, and that it's something that we haven't discussed, but the one of really important point in genomic selection is that it could reduce the time that you need to generate a new population. So that is why when we started to talk about genomic selection, I mentioned that the training population that we use here in Michigan is the F6, but with that information, we are doing prediction in F3s. So in F3s, we are going to start to do crosses to basically stack new or to produce new offspring and stack the good alleles for the traits that we are interested faster. So that it's a really good advantage of genomic selection. And Dr. Crosa from CIMMYT, he mentioned in his talks that genomic selection never gonna beat a traditional breeding in accuracy, but genomic selection can beat traditional breeding per unit of time. So that means that if we are thinking to implement genomic selection, we have to think how we can reduce the cost, that the money that we can use in the breeding program, or that we are using the breeding program, and also we have to think about how we can speed up the process. And we can speed up the process doing predictions in early generations, and try to use those early generations to generate new crosses and new progenies. So that is something that you have to think about genomic selection. And yeah, I think that this formula is really good. Again, if you have questions, just open your microphone. But genomic selection it's not the solution for all the problems that we have in plant breeding, but it's really useful, or seems that it's useful. So a lot of companies, they are using genomic selection to increase the genetic gain per unit of time, and we can if we use the breeder equation, we can see that genomic selection is useful for every single part of that breeding equation. So hopefully you are familiar with this equation, but basically this is the genetic gain, or how we can improve the trait, and that is equal to the selection intensity. So genomic selection can be used to increase the selection intensity, because if we can reduce the first, we can, if you can afford to get the genotypic information and the phenotypic information to create this training population, you can predict the phenotype in a lot of other lines, just if you again can afford to get the genotypic information in other lines. So if you can increase with that strategy, you can increase the size of the population, you can increase with that the selection intensity, because you just are interested to select one percentage of that population. So genomic selection could be useful to increase the selection intensity. As well, to increase the accuracy. Again, I mentioned that genomic selection is not going to be traditional breeding to inaccuracy of selection, but genomic selection can beat it in per unit of time. So something again that we are using here in Michigan is that we have to wait 3 years to take the first data in canning quality and in nutritional value and in yield. But with the models, we can start to do selection in early generation as F3s or F4s. So that means that we are doing selections in early generation when usually we just cannot do those selections because we don't have seed or in the seed is still or the genotypes still have a lot of heterozygous regions in the genome. So we can increase the accuracy of the selection in those early generations because the accuracy was 0 before. And now we are using models to increase or to start to do selections in those early generations. And also we can use we can increase the additive variance in the populations, because with the molecular markers, we can estimate the genetic diversity that we have, and then we can plan ahead, not just to do the crosses to produce new progeny, not just take into account the best genomic value, but also taking into account the lines that have good genomic value or phenotypes, but also have they are not closely related. The genetic relation between them is not really close. So we can again do crosses between lines that are good, but there are some genetic diversity between them. And the last point is that we can reduce the generation interval. So again, as example, what we are planning to do here in Michigan is instead of use F6 or F7s, actually we use F7s or later generations to do the crosses. So we want to start do crosses to produce new progeny in F3s. So that it's gonna speed up that is gonna reduce the generation interval in almost 2 years, and that is going to stack the good alleles faster in the breeding program. So as you can see, with genomic selection, we can increase the genetic gain. That is what we want in the breeding programs. Okay, do you have other questions, thoughts? No. Yeah, and at the end, I want to say that if you are interested in genomic selection in your breeding programs, you have to think about money. That it's one of the most important, I will say, limitations. So if you have to compare what, the sequencing cost of genomic selection versus the conventional breeding that you are using in your breeding programs. For us, genomic selection, we think that is a good investment because the sequencing cost for us here in Michigan is around $10 per sample. It's not cheap, but actually, yeah, it's cheap. Um, yeah, it's cheap. That depends of your country, but here is cheap. And if we compare that with to generate the phenotype, you can see that if we just generate the phenotype in one environment, just one rep, and we take data from minerals and cooking time. So here I just put some cost for in our breeding program. So just to generate this data in one environment, one rep, usually we want to generate that data in more environments, more reps. It's $50. So if you compare $50 versus the $10 that we need to get the sequencing data, so you can see that the difference is important. That is just one genotype, so you just can imagine that usually we plan hundreds of genotypes, so the difference is going to be bigger and bigger. So that is why we really think that genomic selection could be really useful in our breeding program, and that is yeah, it's not nothing actually new. We know that a lot of companies are using genomic selection, but the thing is that we have to start to think how it's the best scenario in our breeding program to implement genomic selection. So that is the challenging part. And if you don't have more questions, it's 4 p.m. In Africa. And I want to finish to say that, yeah, I think that I'm not gonna give you, I'm not gonna record the videos in the hands-on section. So what I'm gonna do is just run the code with you next Thursday. And with that, yeah, in that way you can interact with me if you have questions. And still I'm gonna record that live session to yeah, for you. And I am gonna keep the website for a while. But the code is gonna be available in the website, so you can download the code. Right now, there is one code there. I'm gonna modify it a little bit, that code, because I have to do some comments, and with that, it's maybe easier for you follow the code. But basically, what we are gonna do is use all the theoretical part that we learned. But now in R, we're gonna write the code, and basically we're gonna design alleles with additive effect, with a dominance effect, and with different heritability, so that it just is gonna give you an idea about like why we spent the last 3 weeks learning about quantitative genetics, because that there are a lot of concepts that we need in the breeding program, like in the real life. So we need to understand those basic concepts, and we're gonna predict the phenotype with different heritabilities, and then we are gonna run some basic association analysis to identify the QTL regions that control the traits that where we did the simulations. And at the end, we are gonna run the genomic selection models. And we're gonna use the basic genomic selection model without fixed variables. And then we're gonna start to use the QTL information, or the regions that we identified that have association with the phenotype, as fixed variables to see what happened. And yeah, we are not just gonna, again, simulate the phenotype, but also we're gonna simulate the genotype and the QT or regions that control the phenotype. So I think that that is gonna be fun. But still, yeah, it's really important that you need to have R. So please download R and RStudio, and because yeah, we are just gonna use the software. So if you don't have R and RStudio, you are not gonna be able to run the code. Okay. And yeah, Paul, actually the Paul asked if you are gonna have access to the interactive sessions, and the answer is yes. Actually, the two first sessions are right now in the website. So I'm gonna post this video this afternoon, and again in the hands-on section, I am not gonna post the videos. I am not gonna post more videos for the next week, but yeah, because that was my idea at the beginning. But I think that is gonna be better if I just run the code with you on next Thursday. So be prepared, because maybe, hopefully no, but maybe next Thursday's session is gonna be a little longer. I don't think so, but just be prepared about that. And yeah, and we're gonna run the code together. Basically, and as I'm gonna record the session, so you are gonna have access to do for that. Yeah, later on that. And yeah, if you don't have any questions, we can finish now. And see you next week. And remember, if you have questions after we're done, you can post them in the Slack channel. The Slack channel is for that. Yeah. Okay. Any other question or no?
Thank you.
Okay, guys. So thank you so much for being here, and see you next week. So remember that you are gonna need to have R and RStudio.